Today we will discuss about this MMP9 affects the cell invasion. So how they come up to this conclusion that we will discuss in this video. So they are able to see that whenever the cells are treated with PMA, they shows elevated cell invasion compared to the control case where the cells are not treated with PMA. So here they wanted to find out what in the PMA treated cells cause cell invasion. So in this case what they did is they took the conditioned media of the cells that are treated with PMA, and from the previous experiment that we have also discussed, they are able to see that in the conditioned media there is a increase in the MMP9 levels, and also there is, since MMP9 levels are increased, they are also able to see more fragmentation of the N-cadherin. So now they checked whether these N-terminal fragments that in the previous literature has reported that N-terminal fragment cause cell invasion. So they wanted to check that hypothesis also. So what they did is they first wanted to check whether this N-terminal fragment are present in the extracellular matrix or in the intercellular part of the cells. In order to check that, they divided the samples in between the cell lysate and in the conditioned media. And in the cell lysate, they checked it for the presence of this N-terminal fragment, and also in the case of this conditioned media, they checked the presence of N-terminal fragment. So in the case of cell lysate, almost negligible amount of fragmented N-terminal fragments are present, which is denoted by 90 kilodalton, and most of them are 130 kilodalton. Whereas in the case of conditioned media, they are able to see negligible amount of intact N-terminal fragment. However, the N-terminal fragments are present in an abundant amount in the case of conditioned media, indicating that this N-terminal fragment are being secreted or present in the extracellular matrix rather than in the intercellular part of the cells. After that, coming to the main point, that they want to find out what is causing the cell invasions when they are treated with conditioned media of the cells which are undergone MMP9 exposure. So in order to check that, so right now they have two major candidates. First one is, since the MMP9 level goes up, they think that MMP9 also might cause cell invasions. And second one, the previous literature reported that this N-terminal fragment can cause cell invasions. So they used the antibodies against MMP9 and this N-terminal fragment of this N-cadherin. And also for the control they use IgG. IgG results are shown in the bottom part, but let's just go through in the order. So in this case, these images, in these immunofluorescence images or this macroscopic images, in these macroscopic images they are able to see that when the cells are treated with MMP9 antibodies, they do not show cell invasions, whereas when the cells are treated with N-cadherin antibodies, they are able to show invasions. And similar results they have seen for both the cell lines, it is NPC039 and NPC076. So this indicates that it's the MMP9 that plays a critical role rather than this N-cadherin fragment. And these things they have quantified on the bottom side of the panel B, where the control also shows cell invasions, because in the control case both MMP9 and this N-cadherins are present. And when they used MMP9 antibodies, so it will neutralize the MMP9, and that will leads to the diminishing effect of diminishing cell invasions. And one of the reasons could be that MMP9 does not able to play its role to trigger cell invasions. And in the case where they used N-terminal fragment of N-cadherin antibodies, or this antibodies that target N-terminal fragment of N-cadherin, in that case also they are able to see cell invasions. And this is true for both NPC039 and NPC076, indicating that this N-cadherin N-terminal fragment of N-cadherin does not seem like it plays a role in the cell invasions. After that, they also checked the expression level of MMP9 in the different conditions, and they are able to see that in the case where anti-MMP9 antibodies is used, they are able to see a reduced MMP9 formations, indicating that MMP9 might play a role or might trigger its cell formations. And once these MMP9s are neutralized, they are not able to trigger this MMP9 expression, MMP9 cascade effect, we can say like that. Because once the MMP9s are present in high amount, it will cause more fragment, and that fragmentation of this N-cadherin then helps in the further formation of MMP9. And since in this case where only MMP9 is used, they are not able to only MMP9 is used, and which neutralize the MMP9 that are presently present in the system, and that reduction in the MMP9 will cause reductions in the N-cadherin cleavage and thereby reductions in the MMP9 formations. This is the reason why this MMP9 signals goes down compared to the N-cadherin and control. So finally they conclude that it's the MMP9 that has a major role, that has a major and direct role towards cell invasions. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.